So first page of the guidance says that what is a microcirculation. So definitely microcirculation is all about arterioles, venules and capillaries. Ready? So they start with arterioles, that what is a arteriole. Arteriole is around diameter of 10 to 15 micrometer. Then they divide around 5 to 7 times. Then they turn in a true capillary. So how you can differentiate capillary from arterioles? Arterioles continuously having the muscular layer in their wall, ready, which regulate their size extensively, while capillary doesn't have a muscles in its wall. We will learn the capillary structure later, but on this page they are describing what is metarteriole. Ready? It is a structure which is not having a continuous muscle layer like arteriole but some intermittent muscle, but this muscle are very helpful in constricting and cutting off the tissue blood flow. Okay, you also see a structure which is pre-capillary sphincter. Ready? So how this entire structure helps? So suppose this is not the exact scenario in every tissue. Every tissue somewhat have a different kind of projection of this structure, but ultimately when you are controlling meta-arterioles and pre-capillary sphincter, you can entirely govern the tissue blood flow. Ready? And this structure is nearby the tissue. Fine. So tissue on need, like in hypoxia or nutrition need, excessive carbon dioxide, okay, will act on this meta-arteriole and pre-capillary sphincter and regulate their own tissue blood flow. Okay. Okay. So another thing which we can say about capillary structure is they are made for the exchange. Okay. So ultra microscopic structure, they are also called the exchange vessels. Ready? The ultra microscopic structure is showing that there is a very single layer of endothelial cells. Okay. This kind of capillary seen almost in all body parts, but this is a typical example of muscle and connective tissue capillary wall. Ready? This unicellular This unicellular layer of endothelial cell is surrounded by a thin basement membrane outside the capillary. So you can easily notice here the lake of the muscle wall. Okay. The total thickness of capillary wall is only about 0.5 micrometer. So the average is 4 to 9 micrometer. So you know the diameter of RBC is 7 micrometer. So it is hardly a space left for RBC to squeeze through. So as we move ahead with the guidance, nearby this figure, they have explained the slit pores. Okay. So in capillary, there is a intercellular cleft which is present only at the edge of the cell. Very less area they are occupying. Surface area is one total thousand, only 1,000 of the total surface area of the capillary, but they are extensively helpful for exchange of fluid, water plus water salt ions, ready, for inside and outside of vessel wall. And another thing they are projecting here is this caveoli, ready, caveoli. So they are like cavity like structure in the vessel wall, in this cell wall, inside cell wall of capillary endothelial cell. So scientists are still studying it, but they propose a theory that they helpful in rapid exchange, okay, of the things. They are made of this, the common thing, protein called caveolins, cholesterol and sphingolipids. Ready? They are helpful in endocytosis. So you can see now Then we have moved to another paragraph. This paragraph is talking about the different capillary structure according to different function of a tissue. So definitely brain needs to be protected. So brain capillary have tight junction. Okay, so they are not allowing that much movement. The tight junction are so small they are only allowing the water movements. Okay, which we also call brain blood barrier. So liver is the just opposite. Ready? Liver is our all digestive and nutritive and immunity function, protective function. So liver has to process all the blood. So its capillary junction are so much wide. Okay. The cleft between the capillary endothelial cells are so wide. Ready? So even the plasma protein can pass through. Now we talk about the GIT. So they need to absorb whatever you are eating, they need to absorb. So their intercellular cleft are somewhere midway, not as much large as liver, not small as brain, but somewhere in between. And a very peculiar and a different in a kidney that in midway, not like the cleft, but in midway you find a passage which we will call a fenestra, right? But they are not allowing the passage of a large molecule. They are only allowing the passage of small molecule. Ready? So as you have seen, we have completed that ultra microscopic structure. Now we are moving towards the heading called vasomotion. So like in artery and veins, the movement of blood is not continuous but it is intermittent. We will call it vasomotion. It depends on the contraction of metarteriole and precapillary sphincter. They have given the example how this vasomotion, this intermittent blood flow is regulated. It is mostly on the need of oxygen. Suppose in some portion tissue oxygen decreases, then this meta-arteriole will dilate, the precapillary sphincter will dilate. So intermittent period of this vasomotion, this blood flow will increase and they will remain open for longer time. Okay. So ultimately the concentration of oxygen will rise in that area. So now we are moving forward. You can see here, ready? This is a figure of a capillary and what are these arrows? So this is showing the exchange capacity of capillary for water and nutrients. This depend on a thermal motion, the Brownian movement of water molecule, but water is a so small molecule that when a plasma is passing one time, है ना, प्लाज्मा जब एक बार निकलता है, the water is going in and out 80 times because of its thermal motion. Ready? Very, very fast. Rapid movement of water. Why? To help the transfer of a water soluble substance inside and outside. Okay. So that's why the capillary have a very good exchange between the intercellular area with the intercellular area. Ready? And in intercellular area, it is in very good exchange with cell. So ultimately, there is a good exchange of nutrition, the waste material, the water, carbon, oxygen, carbon dioxide, oxygen with capillary in between capillary and cell. Now the normal rules apply here also like lipid solubility. So if you are lipid soluble, you are passing very fast like oxygen and carbon dioxide. Ready? If you are of a small size, ready? You are passing very fast. So next slide is all about the size and the molecular weight. So the smallest molecule is water. So capillary 4 is around 6 to 7 nanometer and water is 20 times smaller than, so it can pass very fast. So its permeability is like 1. So as you can see, the molecular weight is increasing, the permeability decreasing. So in normally in a skeletal muscle tissue and body tissue, it is not permeable to like plasma protein. Ready? But other tissue like liver, है ना, we need in a digestion and nutrition function, we need a liver. Uh, so its sinusoids are very wide, also allowing the plasma protein to pass. And they are giving now example of kidney here in this paragraph, that it's no doubt its permeability of kidney is 500 times than skeletal muscle, but it is not allowing the plasma protein to pass. Okay. So as we move further, there comes the concept of tissue gel. Ready? On next page, and all this paragraph is about the interstitium and interstitial fluid. So as you can see, we have proteoglycan filament which makes 98% and hyaluronic acid which make 2% of the interstitium. The capillary fluid, water, and which is filtered from the capillary is collected here in interstitial space. Ready? Along with this fiber, it makes a gel-like structure which we call tissue gel. It is almost similar to the plasma except it doesn't have plasma protein because protein is not filtered from the capillary. So as you move ahead in this chapter, you will see these headings: capillary hydrostatic pressure, interstitial fluid hydrostatic pressure, this plasma colloid osmotic pressure, and which proteins are the reason behind it. Interstitial fluid colloid osmotic pressure. So you have to go this, you have to read once this paragraph. They are very easy, you can understand. So when your base is cleared, we will only discuss in class next Monday the Starling forces, which are responsible for exchange between the capillary and tissue. Ready? The Starling equation we will discuss.